Hello friends, today we are going to study about protein function, reversible binding of a protein to a ligand part 2. Myoglobin's oxygen binding curve is hyperbolic. The function of myoglobin depends on its ability not only to bind oxygen but also to release it when and where it is needed. That means to function efficiently, myoglobin has to bind with oxygen also, and it has to release the oxygen also whenever it is needed. What is the function of myoglobin? To transport oxygen from capillaries to muscle tissue. To perform this function, myoglobin has to bind with oxygen at the capillaries and has to release that oxygen in the muscle tissue. Then only it can perform its function. Now, not doing either of the two, that means if it cannot able to bind with oxygen or if it cannot able to release the oxygen when needed, then it cannot function properly. So both the part is important. To bind with the oxygen is also important, and to release the oxygen when needed is equally important. Now this statement we can describe as P, that is protein, plus L, that is ligand, reversibly equal to PL, that is protein ligand complex. That means the reaction should be reversible. Protein should bind with the ligand to give a protein ligand complex, and whenever needed, this protein ligand complex should break down to release the ligand where it is needed. Now, this reaction can be characterized by equilibrium constant Ka. What is Ka? It is association constant. When we are talking about association, so that means the product is protein ligand complex. So that means Ka will be equal to concentration of protein ligand complex divided by concentration of protein into concentration of ligand. So what is Ka? It is an association constant. That means it is the measure of affinity of ligand for the protein. So Ka is a measure of the affinity of the ligand for the protein. More the value of Ka means more affinity. Now, if we rearrange this equation, so what is this ratio? The protein bound with ligand and the free protein. So that means the ratio of the proteins that will be associated with the ligand and the free protein will depend upon the concentration of ligand. More the concentration of ligand means more will be the protein ligand association and less will be the free protein. If the concentration of ligand is less, that means the protein ligand formation will be less and the free protein will be more. Clear? So the ratio of the protein bound with the ligand and the free protein depends upon the concentration of ligand. So PL is directly proportional to the ligand. That means more the concentration of ligand, more will be the protein ligand complex, and less the concentration of ligand means less is the protein ligand complex, or in other word we can say more will be the concentration of free protein. So that means the ratio is proportional to the concentration of ligand. Now, if the concentration of ligand is much more than the ligand binding site, then binding of ligand with protein does not change the concentration of free ligand. That means the concentration of ligand will remain constant. If suppose four ligand binding sites are present and we have multiple ligands, then the binding of these ligands with the protein will not affect the concentration of free ligand. That means the concentration will definitely be affected. If there are 100 molecules of ligand and 4 molecules got attached with the protein, the concentration will definitely decrease. But such a minute difference in concentration will not appreciably change the concentration of free ligand. So, in this condition, we can say that the concentration of ligand remain constant because this much change in concentration is very negligible. So we can say the concentration of the ligand remain unaffected or constant. Now, this condition is broadly applicable to most of the ligands that bind to the proteins in a cell. Now, we have to consider the binding equilibrium from standpoint of the fraction theta. What is theta? Theta is equals to binding site occupied divided by total binding site. So that means binding site occupied means concentration of protein ligand complex divided by total binding site means the binding site in free protein plus the binding site in the bound protein. So this ratio, this fraction is given by theta. In some book, this fraction is also written as YO2. YO2 because we are talking about myoglobin. We'll see in the following slides. So theta is what? It is the fraction. What fraction? Binding site occupied divided by total binding site. So now we have our theta. Now substituting KALP for PL. From where we got this? If you remember, we found association constant is equals to concentration of PL divided by concentration of P into concentration of L. Here, if you take the denominator to the left side, it will become like this. So now, in place of concentration of PL in the theta, we can replace that PL with Ka into concentration of ligand into concentration of protein. So now what will become? Theta is equals to KALP divided by P plus KALP. Wherever in the theta concentration of PL was there, we replaced it with KALP. Now we can take common P. It will become 1 plus Ka into L. Now this P and this P we can cancel out. So now it became KAL upon 1 plus KAL. Now this can also be written as L upon 1 upon Ka plus L. So remember this formula. Theta is equals to concentration of ligand divided by 1 upon Ka plus concentration of ligand. Now we have this formula. Now when we will plot a graph between theta and ligand concentration, we'll get the value of Ka. And how the graph will look like? That means as the concentration of ligand will increase, the fraction will also increase. What is this fraction? Binding site occupied divided by total binding site. As the concentration will increase, this fraction will also increase. The point where it will be 50% saturated. That means 50% of binding sites are now occupied. That where it is, that ligand concentration is known as Kd. So Kd is that concentration of ligand when the fraction is 50%. That is half saturated. What is Kd? Dissociation constant. And it is Kd is equal to 1 upon Ka. So in this formula, now in place of 1 upon Ka, we can write Kd. So the new formula will become theta is equals to concentration of ligand divided by Kd plus concentration of ligand. Clear? What is Kd? Kd is that concentration of ligand when the fraction is half saturated. When 50% of protein is bound with the ligand. What is Kd? It is the concentration of ligand only. Right. So what will be the unit of Kd? Whatever is the unit of ligand concentration, that unit will be the unit of Kd because Kd is that ligand concentration. So this theta formula in place of 1 upon Ka because dissociation constant is inversely proportional to association constant. So in place of 1 upon Ka, we can write Kd. So now we have theta is equals to ligand concentration upon ligand concentration plus Kd. What is Kd? It is that ligand concentration when theta, that is fraction, is 50% or half saturated. Or we can say half of the binding sites are occupied. So that means when concentration of ligand is equals to Kd, half of the binding sites are occupied. What if concentration of ligand is less than Kd? That means less protein is bound with the ligand. So this is less. Because Kd is talking about dissociation constant, that means If the value of Kd is less, the affinity is more because association constant and dissociation constant are inversely proportional. So if the Kd value is low means affinity is more, and if the affinity is more means less ligand concentration will be required to reach the saturation. Now, as our protein is myoglobin and our ligand is oxygen. Oxygen is a gas. Therefore, we can rewrite the formula of theta. What is our formula of theta? We can rewrite it as, because ligand is oxygen. Now, what is Kd? It is that ligand concentration when the fraction will be 50%. That means half saturation. So now we can write as, that means it is that concentration of oxygen when half ligand binding site is occupied. Now, because in case of gas, it is easy to measure their concentration as a partial pressure. So now we can rewrite the same formula. Theta is equals to partial pressure of oxygen. Why? Because oxygen is a gas. So it is easy during the experiment to measure the concentration of oxygen in terms of their partial pressure. So the same formula now will become, wherever oxygen concentration is there, we will write partial pressure of oxygen plus P50. What is P50? When 50% fraction or 50% ligand is associated with protein or 50% ligand binding site is full. So that is O2 0.5, and from where it came, it is nothing but the Kd. The same formula, if you trace back, you can find P50 in place of O2 0.5, O2 0.5 in place of Kd. Now graphically, this can be plotted as hyperbola curve. Here, if you remember the earlier curve, we have written here ligand concentration. So ligand here is oxygen concentration in terms of partial pressure. Here it was theta. That time I told you that in place of theta in some book we find it as written as YO2 and the hyperbola. So what the hyperbola is saying, as the concentration of oxygen will increase, the fraction will increase. When the fraction is at 50% or the protein is half saturated, this value is Kd, right? Or we can say it is P50. This value is what? P50. Now, what else this hyperbola curve explains to us? You can see in the curve, first of all, there is a steep rise and then it slows down and become static. That means when the concentration of oxygen is increasing, it will start binding with the protein. And finally, when all the proteins have been bound with the oxygen, after that, further increase in oxygen concentration will not increase the fraction because now no binding sites are available for binding more ligand. So there is a steep increase in the concentration. That means whenever till the binding sites are available, as the concentration of oxygen will increase, it will go on binding with the protein. Once all the binding sites are occupied, it will become static. This is the first thing. Second thing is such type of curve represents the independent binding of ligand to the protein. That means the binding of one ligand to the protein will not affect the binding of other ligands. Why it is happening? Because myoglobin is having a single subunit. Therefore, the binding of ligand with one myoglobin, how come it will affect the binding of ligand with other myoglobin? Because all are separate. So that means such type of hyperbolic curve will observe in all the cases wherever the binding of one ligand will not affect the binding of another ligand. All are independently binding. Nobody is affecting the other's binding. Correct? So it happens in all the single subunit protein and all the multimeric proteins where subunits do not communicate. So the binding of ligand to one subunit will not affect the binding of ligand to the other subunit. This will only happen if the protein is single subunit because all the ligands are binding with the separate protein, and also to those multimeric protein where the subunits do not communicate with each other. So that's why the Myoglobin binding curve is hyperbolic in nature. It is a hyperbola curve. Now, one question from my end for you all. At what oxygen concentration will myoglobin be 75% saturated with oxygen? That means you have given the fraction and you have to calculate the oxygen concentration, that is PO2. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe and stay happy.